Okay, so again, with these simple in vitro assays, it's of course important to do validation experiments. So in other experiments, we showed that we could recapitulate the results in a standard immunoprecipitation experiment, in vitro pull-down experiment. We could get the same result if instead of probing with this little PhD domain, we probed with full-length air. We could show in chromatin immunoprecipitation experiments that air was physically associated with hypomethylated H3K4 residues. And data both from our lab and the Pedersen lab, if one does whole genome mapping, what is found is that air-induced genes are enriched in chromatin that has hypomethylated H3K4 residues. So what I mean by that is if you look at the air-induced transcripts in a cell that's not expressing air, what you see is these are in hypomethylated chromatin. So again, I don't have time to show you any of this, but what I did want to do was show you what I call the bottom line experiment. And so what we did here was take the NMR structure of this PhD domain of air, model it together with the histone 3, unmethylated histone 3, and find this D299 residue, which we thought would be very important because it makes this bridge here. And what we could show, in fact, was that this 299A mutation abrogates the ability of air to bind to H3K4 at all. So then what we did was to make transgenic mice that expressed either only wild-type air or only air that has this single point mutation. And when we get these mice and isolate their medullary epithelial cells and do gene expression profiling, what we're comparing here is expression of the individual genes in the air wild-type transgenic versus the knockout, and the air mutant transgenic versus the knockout. And in red are air-induced genes in a wild-type situation, and in blue are the air-repressed genes. And if they were expressed the same in the wild-type or knockout, they'd be along this diagonal. The fact that they're splayed along the x-axis here means that the majority of them, while being upregulated in wild-type, are poorly upregulated in the knockout. And then finally, probably the most important point, is that we can, as I mentioned, air knockout mice have a multi-organ autoimmune disease. And if we make mice that have only this single point mutation, we perfectly recapitulate the phenotype of the total air knockout mouse. So that's whether you look at survival, whether you look at autoantibody production, or whether you look at infiltrates. So we do think that this is an important interaction with air that's involved with its targeting to particular loci. Now, for the rest of the time, I'd like to talk about another complex here because we realized that these proteins are well known in two other molecular contexts. So, but to validate their importance, the first thing that well, I should say, I'll tell you the bottom line in case things get a bit rushed at the end, is that we believe that air's participation in this complex not only refines its targeting to particular loci, but also promotes transcriptional elongation. Okay, but again, to validate these complexes, we did, first of all, a series of immunoprecipitation experiments where instead of precipitating with air, we precipitated with all the other members of the complex and looked to see what came down with it. And basically, if you precipitate with DNA PK, all these friends come down. Same if you precipitate with Topo. And so we could validate all the members like that and add some additional members like H2AX and Ku. We could also do SHR or siRNA knockdown experiments where we could knock down DNA PK and it knocked down the ability of these to interact with each other. And we also did a series of experiments like that. But perhaps most important is to know whether or not mutation or removal of these proteins has an ability on air to induce peripheral tissue antigen transcripts in medullary epithelial cells. Now, many of you will recognize that DNA PK, the catalytic subunit of DNA PK, is the protein that's mutated in the SCID mutant mouse. So we had that as a nice way to be able to look at the effect on air-induced transcripts in the thymus. Now, we couldn't do the simple experiment of just looking to see, just comparing what peripheral tissue antigen are expressed in medullary epithelial cells of wild type and air knockout and SCID mice, because SCID mice have a rudimentary thymus, and that's because there is crosstalk between T cells and stromal cells in the development of the different epithelial cell compartments of the thymus, in particular the medullary epithelium. So what we needed to do then was to do a reconstitution with wild-type bone marrow. And so what we end up having are mice which are either SCID mice or RAG knockout mice, which are reconstituted with bone marrow from wild-type mice so that they'll reconstitute the thymus, and you see that this does happen. And so then we can look just to see whether a mutation in SCID versus a mutation in RAG has an effect on peripheral tissue antigen expression. And so what we find is that there's essentially no difference between the RAG, reconstituted RAG, and the reconstituted SCID mouse in the expression of air. They're the same. However, if we look at the peripheral tissue antigen genes, and here we have a panel of them here, we see that while RAG is not much affected, each one of these is rather strongly affected in the SCID mutation. Not as strongly affected as in the wild type, but still strongly affected. And I won't show you the data, but we confirm this in a broader scale using microarray analysis. So DNA PK is important for this thymic transcripts. And we also thought TOP2A presented a nice possibility to validate the importance of these complexes, not because of the knockout, because that's too drastic, but rather because there is a whole series of inhibitors of TOP2A which impact at different points in TOP2A's activity. So as you know, TOP2A is required to relieve torsional stress as transcription and replication is taking place. So TOP2 makes a double-stranded break and then forms a covalent bond on each one of the strands. One strand passes through the other, and then TOP2 closes it up. And as I mentioned, then there are inhibitors that impact at different points. So for example, aclarubicin impacts at the point of TOP2 binding to the DNA itself. And we could show, here I show one example, but we have many examples, where aclarubicin specifically inhibits the ability of air to induce different peripheral tissue antigen transcripts in transfected cells. And here we're looking at this KRT14, but as I said, it's true of many other transcripts. So that fit very nicely. And then Jacob continued these studies and came up with what I think was an absolutely amazing result with a different TOP2 inhibitor, and this is etoposide. So etoposide doesn't inhibit the binding of TOP2 to the DNA. It does bind, but it's what's called a TOP2 poison because it freezes the covalent TOP2 DNA complex and stabilizes double-stranded breaks, or actually can either single-stranded or double-stranded breaks. Okay, so being the excellent postdoc that he is, Jacob did a very well-controlled experiment where he took a bunch of different air-induced transcripts, known to be the most induced on your left, and the lesser induced on your right. And for each one of these, he had 4 different cell samples. One was not treated or transfected with anything. Another was just treated with etoposide. Another was just treated with air. And the last one was treated with both air and etoposide. And what you can see is that there is an amazing concordance between induction of these transcripts by etoposide and induction by air. And that when you add the two together, you don't get any increase, so it's not synergistic, but rather suggesting that they're in the same pathway. So that was interesting, but again, we wanted to do it on a more global scale, so we did gene expression profiling, comparing etoposide-treated cells. Here I'm showing 293 cells, but we have the same results with medullary epithelial cell line, comparing etoposide-treated with air transfected. And what we find is that, here what we have in black is the data on the etoposide-treated cells, and then in red we have the known air-induced transcripts. And what you can see is that there is a correspondence between what's induced by air and what's induced by etoposide with a p-value of 10 to the minus 128. So we thought this was probably true. And actually, you also have, even with the air depressed transcripts, it's also highly significant, although not as striking. So this is not just a nonspecific response of anything that causes double-stranded breaks, because if we look at a series of other agents that introduce double-stranded breaks by different methods, this doesn't happen. It's only with etoposide. So as I mentioned, etoposide is a topo poison, which freezes this complex and stabilizes DNA breaks. So we were wondering whether air might not be an etoposide mimic. And so we did a classical kinetoplast in vitro assay. So kinetoplast DNA is just a series of concatemers, and they run very high on the gel. If you decatenate, then it runs like this. And indeed, if you treat these with topo, you see that this high molecular weight band comes down. And if you treat with either air or etoposide plus topo, you get a nice enhancement of this in vitro assay. And then, given that result, the prediction would be that the medullary epithelial cells in mice that have air would have more double-stranded breaks than the medullary epithelial cells in mice that don't have air. And as I mentioned, we found that H2AX was a member of the complex that we were interested in, so we could use gamma H2AX as a label for double-stranded breaks and actually do flow cytometry. And what we could find when we isolated the medullary epithelial cells, that there was always between a 3- and 5-fold increase in the number of double-stranded breaks in mice that had air versus those that did not.